The overall goal of this methodology is to fabricate an antibody array with applications in single-cell analysis.
This method can help answer key questions in the field of single-cell analysis, such as how can we detect secret proteins and intracellular proteins simultaneously from the same single cell, and how can we fabricate a microarray platform to accomplish that objective.
The main advantage of this technique is that it is customizable. By varying the flow patterning strategies, arrays of various dimensions may be created.
In advance, generate the SU8 master for the barcode flow patterns. Instructions are provided in the text protocol. This involves designing and making a chrome photomask. Placing the mask on a photoresist coat and applying UV provides the pattern. To start preparing the PDMS barcode mold, combine 40 grams of silicone elastomer base with 4 grams of curing agent and stir vigorously for 10 minutes. Then degas the prepolymer solution for 20 minutes under vacuum. Next, load the solution into a petri dish containing a silicon wafer with the SU8 master of the barcode pattern. Pour the solution to a depth of 7.45 millimeters. Then, degas the mixture in the dish to remove any remaining bubbles. Follow the degassing by baking the mixture for an hour at 75 degrees Celsius to cure the PDMS. Next, cutting with a scalpel, carefully remove the barcode mold. Then, carefully peel the slab from the wafer.